大家好，我是 Jenny， 欢迎来到沉思者 J。 有一种天才，是在寂静中燃烧的，没有掌声，没有舞台，没有同行的认可，只有一个人，一片玉米田，和一个后来改变整个生物学的发现。今天我们要说的故事，是一位科学家，他花了30年，在无人相信的荒野中独自前行，最后用一座诺贝尔奖，让全世界欠他一个迟到的道歉。他叫芭芭拉麦克林托克，遗传学史上最孤独的革命者。让我们从他的童年说起。1902年6月16日，芭芭拉出生在美国康乃狄克州的哈特福，她排行老三，上面有2个姐姐，下面有1个弟弟。父亲是一位医生，母亲是位有艺术气质的女性。家里的氛围既严肃又充满好奇心。芭芭拉从小就和别的孩子不一样，她特别享受独处，喜欢一个人安静的思考。她不是那种喜欢和同学嬉闹的孩子，更愿意一个人坐在角落，沉浸在某个问题里面，可以好几个小时不说一句话。母亲曾对亲友说过，这孩子天生就需要空间，你管太多，他反而走不远。这句话放在整个人生的尺度上看，简直像是一个预言。因为芭芭拉的父亲工作关系，他在婴儿时期就和父母分开，在外祖父母家住了好几年。这段独立的童年时光，或许让他很早就学会了和自己相处，也学会了在没有人陪伴的情况下，自己找到方向。后来全家迁居到纽约布鲁克林，芭芭拉在那里完成了他的中学教育。他对学校的科学课着迷，对什么事情都想问个为什么。他不满足于课本上的答案，他要的是更深的解释，更根本的原理。高中时期他就展现出惊人的专注力，一旦开始研究一个问题，就可以几个小时不动，完全沉浸在自己的世界里。他的母亲其实有些担心他太过投入学术。在那个年代，女孩子读太多书被认为不是一件好事，甚至可能会影响找到好人家。但芭芭拉根本没有在想这些，她一心想进大学念科学。1919年，芭芭拉进入康奈尔大学农学院就读，这个选择在当时相当罕见。农学院在那个年代几乎是男性的天下，但康奈尔的农学院比较特别，它是少数愿意招收女学生的学系之一。芭芭拉毫不犹豫地选了遗传学为主修。那个时候她遇到了改变她一生的老师，遗传学家克拉伦斯·哈钦森。他看出了芭芭拉非凡的潜力，特别邀请这个大学部的学生加入研究所的遗传学课程。在当时，这是非常破例的待遇，一个大学生坐在研究生的课堂里，和比他高1个层级的学生一起上课。但芭芭拉不只跟上了，他还让那些研究生印象深刻。芭芭拉就这样进入了遗传学的世界，然后就再也没有出来过。康奈尔的玉米田成了他一生的舞台。那个年代的遗传学正在经历一场奇特的革命。孟德尔的遗传定律已经被重新发现，托马斯·亨特·摩根的果蝇实验正在慢慢揭开基因的秘密。科学家们开始相信，遗传的资讯是沿着染色体线性排列的，但当时还有很多谜团没有解开。遗传重组是怎么发生的？基因是怎么从亲代传到子代的？染色体的物理结构和遗传行为之间究竟有什么关系？芭芭拉选择了玉米作为她的研究材料，这个选择非常聪明。玉米的染色体又大又清晰，在显微镜下看得一清二楚，而且玉米的种子外表颜色可以直接反映基因的状态，是一个绝佳的遗传研究材料。芭芭拉的显微镜技术举世无双，它可以辨识出染色体上极细微的特征，区分出不同染色体的形态差异。这个能力在后来的发现中起了决定性的作用。1927年，芭芭拉拿到了遗传学博士学位，从此开始在康奈尔展开她的研究生涯。她的研究成果来得很快，在1930年代初期，她和同事哈利叶特·克雷顿做出了一个重要的实验， 用玉米染色体直接证明了遗传重组确实发生在染色体的物理交换上。在这个实验之前，遗传重组只是一个理论上的推论。科学家们认为，亲代的基因在传递时会发生混合，但没有人亲眼看见过这件事是怎么发生的。芭芭拉和克雷顿的实验改变了这一切。他们追踪了特定的染色体标记，清楚地看到了染色体的某一段在遗传的过程中发生了实际的物理交换，不是理论，是实际看得到的物质变化。这个实验被称为遗传学史上的里程碑。摩根在收到这篇论文的预印本之后，催促他立刻发表，因为有另一组科学家也快要得出同样的结论。两人在1931年发表了这个成果，确立了染色体与遗传重组之间的直接关系。这篇论文至今仍是遗传学的经典文献之一。这一年，芭芭拉只有29岁。接下来几年，她继续在染色体研究上做出重要贡献。她和同事马科斯·罗德斯一起绘制了玉米的染色体图谱，建立了玉米染色体上各个基因的相对位置。这是当时最完整的植物染色体图谱之一。他还研究了染色体末端的特殊结构，发现了一种他称为端粒的结构，可以保护染色体末端不发生融合或降解。这个发现在几十年后又重新受到重视。今天我们知道，端粒在细胞老化和癌症中扮演着关键角色。研究端粒的科学家最终也在2009年获得了诺贝尔奖。按照正常剧情发展，这样的科学家应该很快就会在大学找到教职，然后一步步成为领域的顶尖人物。但现实不是那样的，因为她是女性。在那个年代的美国学术界，女性科学家几乎找不到稳定的教职，顶尖大学的科学系几乎清一色是男性教授。女性就算做出再好的研究，也难以跻身正式的学术职位。芭芭拉在康奈尔当了好几年的研究员，做着让同事赞叹的研究，却始终没有正式的教授职位。他申请过几所大学的职位，要么石沉大海，要么被委婉地告知这个位置不适合女性。他后来也在密苏里大学担任过助理教授，虽然在那里继续了他的玉米研究，但大学的氛围让他感到格格不入。他不擅长处理人际关系的复杂，他只想做研究。1941年，他加入了冷泉港实验室，位于纽约长岛的这个研究机构给了他一个能够安心做研究的地方，没有繁重的教学义务，没有细琐的政治，只有他和他的玉米田。冷泉港成了他后来数十年的家。在这里，他继续研究玉米，一行一行地种，一季一季的观察，年复一年。在那片玉米田里，他和自己的研究建立了一种近乎亲密的关系。他后来曾说过，每一株玉米都有自己的个性，他能感觉到它们。这不是玄学，这是长期深度观察所带来的直觉。当你花了足够长的时间凝视一个东西，你的大脑开始在意识层面以下做判断。有些研究者把这称为直觉，但其实这是高度积累的模式识别能力。就在这样的状态下，他看见了其他人都没有看见的东西。1944年到1950年之间，芭芭拉在玉米的染色体上发现了一个完全出乎意料的现象。玉米的种子有时候会出现奇怪的颜色斑点，紫色的底色夹杂着白色或橘色的色块，图案千变万化，完全不遵守孟德尔的遗传规律。按照孟德尔的定律，显性和隐性基因的表现应该有规律可循，但芭芭拉看到的这些种子，就像是有人在染色体上胡乱涂鸦了一样，完全不按牌理出牌。一般的科学家看到这个，可能会觉得只是杂讯，或者实验材料不纯，换一批种子重做实验就好了。但芭芭拉不这样想，她仔细追踪每一粒种子的清代资讯，记录每一株植物的遗传背景，一一对照显微镜下的染色体变化，花了整整6年的时间，得出了一个让自己都难以置信的结论：基因会跳动。是的，基因可以从染色体的一个位置跳到另一个位置。 这不只是理论上的猜想，芭芭拉有完整的染色体追踪记录，每一个步骤都有显微镜观察作为佐证。她做了大量的杂交实验，追踪了几十个世代的种子，每一个数据都精确到染色体上的具体位置。她把这种可以移动的基因元素称为转位子，并且发现了2种相互作用的元素，一个负责控制，一个负责移动。她叫它们控制因子与解离因子。更重要的是，她发现这些移动的元素会影响周围基因的开启和关闭。当一个元素跳进某个基因附近，那个基因的表现就会改变。这个发现的意义极为深远。如果基因不是固定在染色体上的，那么整个关于遗传稳定性的假设就需要重新思考。基因的表现可能受到更复杂的调控机制影响，这在今天听起来理所当然，但在1950年，这简直就像是有人说地球是圆的一样颠覆。那个时代的主流观念是，基因是稳定的，是固定在染色体上的，每个基因有自己的位置，就像书架上有标号的书，不会自己跑来跑去。芭芭拉说，书自己在动。1951年，芭芭拉在冷泉港的年度研讨会上做了一场报告，把她的发现告诉同行。她准备得非常充分，带来了详细的数据、完整的染色体图，以及她6年来的观察记录。结果几乎是一片沉默，少数几个人提出了质疑，多数人则是困惑地离开。有人认为她的数据有问题，有人认为玉米和其他生物不能类比，还有人根本不相信基因会移动。据说当时有位在场的科学家在报告结束后对同事说，这个女人是不是搞错了什么？整个1950年代，芭芭拉的理论几乎无人接受。她尝试把研究写成论文投稿，收到的回应要么是困惑，要么是拒绝。她甚至停止了在重要期刊上发表相关论文，因为每次送出去都石沉大海。一个人把自己最重要的发现说出来，然后被整个世界无视，你能想象那是什么感觉吗？芭芭拉没有崩溃，她继续种她的玉米，继续做她的研究，继续记录她的观察。她后来曾说，如果你知道自己的研究是真实的，外界的声音其实不影响你，你只是继续做你应该做的事。重要的是研究本身，不是别人现在怎么看你。如果你的研究是真实的，它早晚会被看见。这句话说起来容易，做到需要非人的意志力，但芭芭拉做到了。她在冷泉港的那栋小屋里，继续过着她自己的生活，早起种玉米，下午在显微镜前工作，晚上阅读和思考。就这样年复一年，她的生活极为简单，没有多余的社交，没有多余的消费，她不需要太多，她有玉米田和显微镜就够了。这种生活方式在外人眼中或许是孤僻，但在芭芭拉自己的感受里，那是一种完整的状态。她喜欢她的工作，她相信她的工作，这就够了。1950年代末到1960年代，分子生物学的时代来临了。科学家们开始在细菌和病毒上研究基因。法国科学家贾克·莫诺和弗朗索瓦·贾科布在大肠杆菌上发现了基因调控的机制，也就是著名的操纵子模型。他们发现基因的表现可以被其他分子开启或关闭，这个调控机制非常精妙。有趣的是，他们的发现和芭芭拉的控制因子理论有着惊人的相似性。莫诺和贾科布后来因为这个发现，在1965年获得了诺贝尔奖，但那个时代的分子生物学家还是没有连接到芭芭拉的工作，因为他研究的是玉米，而他们研究的是细菌。学界的壁垒让两个平行的发现在很长一段时间里各自孤立。到了1960年代末，情况慢慢开始改变，科学家们在更多生物体上陆续发现了可以移动的基因元素，果蝇、酵母菌、细菌，甚至哺乳动物。人们开始回头看芭芭拉在20年前的研究，然后开始意识到它是对的。 这种可以移动的遗传元素，后来被统称为转位子，或者更口语的叫法，跳跃基因。1970年代，分子生物学工具越来越精进，科学家们可以直接在 DNA 序列层面观察和追踪转位子的移动。芭芭拉从显微镜下观察到的现象，现在终于有了分子层面的直接证据。学界开始正式承认，芭芭拉在1950年代说的那些话是正确的，不只是正确，而且是革命性的。今天我们知道，跳跃基因普遍存在，所有已知生物的基因组里都有它们，包括人类。人类基因组中，大约有 45% 的序列来自于各种转位元素，它们不只会移动，还在生物演化中扮演了重要的角色，影响基因的表现，推动物种的演化，甚至参与了免疫系统的多样化。科学家们还发现，转位子在某些癌症的发生中扮演了角色，在某些遗传疾病中也有关键作用，这让它成为今天医学研究的重要课题。芭芭拉在1951年说的那些话，需要30年才被世界完全理解。1970年，芭芭拉获得了美国国家科学奖章，那是美国政府给予科学家的最高荣誉之一，由总统亲自颁授。1971年，他当选美国国家科学院院士。1981年，他获得了麦克阿瑟奖，也就是俗称的天才奖。这个奖每年只颁给少数人，专门给各领域表现卓越的杰出者，附带一笔可观的无条件资助金，让获奖者能够自由追求自己的工作。芭芭拉的名字终于开始被广泛提及，科学界开始重新审视他这几十年的工作，越来越多的人意识到他的贡献有多么深远。1983年，诺贝尔生理学或医学奖颁给了芭芭拉麦克林托克，理由是发现可移动的遗传元素。她是那一年唯一的获奖者，也是第一位单独获得这个奖项的女性。那一年，她已经81岁了，距离她在1951年做出那场无人理解的报告整整32年。诺贝尔委员会打电话通知她的时候，她正在冷泉港的院子里捡胡桃。她接到电话，确认了消息，然后说了句好的谢谢，就继续去捡她的胡桃了。这就是芭芭拉麦克林托克。得奖之后，她接受了很多采访，记者和科学史学家蜂拥而至，每个人都想知道这30年间被忽视的感受是什么。她的回答让很多人惊讶。她说，我从来不觉得被遗弃，我只是做我觉得应该做的事，获得认可或不获认可都不影响研究本身的意义。有记者问她，是否对那些当年忽视她的科学界感到愤恨。她说，愤恨什么？他们只是还没准备好理解而已。每个时代有自己的限制，那不是任何人的错。她说，如果你爱你的工作，你就是幸运的人，因为你已经得到了奖赏。外部的认可只是附加的，不是核心的。这种超然，不是冷漠，而是真正的热爱所带来的自由。当你的快乐不依赖别人的掌声时，你就很难被打倒。芭芭拉拿到诺贝尔奖之后，生活方式几乎没有改变，她还是住在冷泉港的同一栋小屋，还是每天去看她的玉米田，还是在显微镜前工作，还是早睡早起。有人问她，得了这么大的奖，有没有什么梦想想要实现？她说，我只希望能继续做研究。她确实继续做了，一直到她晚年。1992年9月，芭芭拉麦克林托克在纽约辞世，享年90岁。她从未结婚，没有孩子，她的一生就是她的研究。但说她的一生只有研究，其实是一种误解。芭芭拉有非常深厚的友谊，她和几位同行保持着终生的友谊。她喜欢音乐，喜欢大自然，喜欢在冷泉港的海湾边散步。她对人生有很深的哲学思考。她曾说，生命中最重要的事是找到你真正在乎的东西，然后对它忠诚，不管外面发生什么。她留下的 是一个持续延伸的理论基础，至今仍影响着整个科学界。跳跃基因的研究，至今仍是遗传学和演化生物学的核心课题。它的研究直接影响了我们对基因组稳定性的理解，对演化机制的认识，对癌症和遗传疾病的研究，对基因编辑技术的理论基础。可以毫不夸张地说，今天所有研究基因组的科学家都站在芭芭拉的肩膀上。从她的故事里，我想留给大家一句话：真正的工作不需要观众，只需要你自己和那片玉米田。如果你今天也在做一件没有人理解的事，想想芭芭拉，继续种你的玉米吧。感谢你今天陪我一起认识这位了不起的大师，我们下次见喽。Hello everyone, I'm Jenny. Welcome to Thinker J. There is a kind of genius that burns in silence. No applause, no stage, no recognition from peers. Just one person, a cornfield, and a discovery that would eventually change the entire field of biology. Today we are sharing the story of a scientist who spent 30 years walking alone in a wilderness where no one believed her. Then, finally, a Nobel Prize arrived to deliver the apology the world had owed her for decades. Her name was Barbara McClintock, the loneliest revolutionary in the history of genetics. Let us begin at the very beginning. Barbara was born on June 16, 1902, in Hartford, Connecticut. She was the third of 4 children, with 2 older sisters and a younger brother. Her father was a physician and her mother was a woman with strong artistic sensibilities. The atmosphere in their household managed to be both intellectually rigorous and genuinely curious about the world. From a very young age, Barbara was simply different from other children in her orbit. She loved being alone more than almost anything else she could imagine. She preferred sitting quietly by herself, turning a problem over slowly in her mind, to any kind of group activity or social interaction. She was not the child who enjoyed running around with classmates or filling her days with noise and company. She would much rather find a quiet corner somewhere and become completely absorbed in something that genuinely interested her, sometimes for hours at a time, without saying a single word to anyone around her. Her mother once told friends and relatives that this particular child was born needing space. If you hover over her too much, she will never go far. Viewed through the lens of an entire lifetime, that remark sounds almost uncannily like a prophecy. Because of her father's professional obligations, Barbara was actually separated from her parents as a very young infant and spent several formative early years living with her grandparents. That early period of relative independence may have taught her, at a very young age, how to be genuinely comfortable in her own company and how to find her own direction without waiting for someone else to show her the way. This is not a small thing. Learning to navigate the interior landscape of your own mind, without constant external input or reassurance, is a skill that most people never fully develop. Barbara had it from childhood, and it would sustain her through decades of professional isolation that would have broken most people. Later the entire family relocated to Brooklyn, New York, where Barbara completed her secondary education. She was deeply fascinated by her science classes. She wanted to ask why about essentially everything she encountered. She was never satisfied with the surface answers provided in textbooks. She wanted deeper explanations, more fundamental principles, the actual mechanisms behind the phenomena she was studying. During high school she already demonstrated a remarkable and unusual capacity for sustained concentration. Once she began working through a problem that genuinely interested her, she could remain completely still for hours, fully immersed in her private intellectual world, entirely unaware of time passing. Her mother was, honestly, somewhat concerned by all of this. In that era, a young woman who was too intensely focused on academic work was not generally considered admirable or practical. Such a disposition was thought to be potentially counterproductive for a woman's social prospects. These were the unspoken calculations that governed expectations for young women at the beginning of the 20th century. But Barbara was not thinking about any of that. She had already decided she wanted to go to university and study science. The social expectations of her era were simply not a category that held much weight for her. In 1919, Barbara entered the College of Agriculture at Cornell University. This was a genuinely unusual choice at the time. Agriculture was almost entirely a male domain in those years. But the Cornell College of Agriculture was one of the few academic programs that actually admitted women, and so that was where Barbara went. She chose genetics as her major without any hesitation or uncertainty. There she encountered a teacher who would fundamentally alter the direction of her life. The geneticist C.E.B. Hutchison recognized Barbara's extraordinary scientific potential early on and made the exceptional decision to invite this undergraduate student to join graduate-level genetics courses. In that era, this was a genuinely remarkable privilege. An undergraduate student sitting in the same room as people who were operating at a higher academic level. But Barbara did not merely keep pace with those graduate students. She made a real impression on them. They could see, quite quickly, that this person was operating differently. And so Barbara entered the world of genetics. She would never really leave it. The cornfields of Cornell became the stage for the rest of her life. Genetics in those years was passing through a period of profound and exciting transformation. Gregor Mendel's laws of heredity, which had been largely overlooked for decades after his death, had been rediscovered and were being integrated into a broader scientific framework. Thomas Hunt Morgan's landmark experiments with fruit flies at Columbia University were steadily revealing the hidden architecture of genes and their positions on chromosomes. Scientists were growing increasingly confident that genetic information was arranged in a specific linear sequence along chromosomal structures. But enormous and fundamental mysteries still remained entirely unresolved. Exactly how did genetic recombination actually occur at the physical level? How were genes reliably transmitted from one generation to the next? What was the precise mechanical relationship between the physical structure of chromosomes and the statistical patterns of inheritance that Mendel had first described? Barbara chose corn as her primary research organism, and this was a particularly astute choice. Corn chromosomes are unusually large and morphologically distinct, which makes them clearly visible and identifiable under a standard research microscope. The color patterns on individual corn kernels can directly and visibly reflect the state of specific underlying genes. This made corn an exceptionally transparent window into genetic processes. Barbara's skills with the microscope were in an entirely different category from those of most of her contemporaries. She could identify extremely subtle structural features on individual chromosomes. She could reliably distinguish between fine morphological differences that others would routinely miss. She could recognize individual chromosomes by their specific physical characteristics, almost the way you might recognize old friends by their faces. This ability, developed slowly and deliberately through years of patient and meticulous observation, would eventually prove absolutely decisive in her most important work. In 1927, Barbara completed her doctorate in genetics and began building her independent research career at Cornell. Her findings came quickly and with considerable force. In the early 1930s, working in close collaboration with her colleague Harriet Creighton, she designed and executed an experiment that would become one of the defining landmarks in the history of the field. Using corn chromosomes directly as their experimental material, they provided the first concrete physical proof that genetic recombination actually occurs through a genuine physical exchange of chromosome material. Before this experiment, genetic recombination was only a theoretical inference. Scientists had good statistical reasons to believe that parental genes mixed and recombined during transmission. But no one had ever directly observed the physical mechanism that made this possible. The idea existed at the level of logical deduction, not direct empirical evidence. Barbara and Creighton's experiment changed all of that in a decisive way. They designed a clever system using corn plants with chromosomes that carried distinctive physical markers visible under the microscope. By tracking these markers through generations of controlled crosses, they could directly observe whether chromosome segments had physically exchanged between parental chromosomes. What they saw was unambiguous. A specific segment of a chromosome underwent an actual physical exchange during the inheritance process. Not theory. Not inference. Not a statistical pattern. A real, observable, material event, visible through the microscope. This experiment is still widely considered one of the genuine milestones in the history of genetics. Thomas Hunt Morgan himself, upon receiving an early copy of the paper, pressed Barbara urgently to publish immediately. Another research group was moving toward the same conclusion, and priority mattered. Barbara and Creighton published their results in 1931, definitively establishing the physical basis of genetic recombination. That paper is still read and cited as a classic of the field, nearly a century later. That year, Barbara was 29 years old. In the years that followed, she continued generating important contributions to chromosome biology. Together with her colleague Marcus Rhodes, she constructed a detailed map of the corn genome, establishing the relative positions of genes across all 10 corn chromosomes. This was one of the most complete and accurate plant chromosome maps produced up to that point. She also turned her attention to the peculiar structures at the very ends of chromosomes. Through careful observation, she identified what she described as a telomere, a structure whose function was to protect chromosome ends from degrading or fusing inappropriately with other chromosomes. This was another insight that was largely set aside for decades, then rediscovered much later and recognized as profoundly important. Today we understand that telomeres are central to cellular aging, to cancer development, and to fundamental aspects of how cells divide. 3 scientists who devoted their careers to studying telomere biology received the Nobel Prize in Physiology or Medicine in 2009. Following the normal academic trajectory for a scientist with this record, Barbara should have quickly secured a permanent faculty position at a leading research university and risen steadily to prominence. But reality had different plans. Because she was a woman. It is worth pausing here to understand what that actually meant in practical terms. The exclusion of women from permanent academic science positions in mid-20th century America was not a matter of formal policy at most institutions. It was something more pervasive and in some ways more difficult to fight, an unspoken consensus about who science was for and who was naturally suited to lead it. Women could work in science. They could be research assistants, laboratory workers, or in some cases visiting researchers. But the tenured professorships, the department chairs, the laboratory directorships, the positions that came with real resources and institutional standing were almost exclusively reserved for men. This was treated as natural, as simply reflecting where talent and ambition happened to reside. For Barbara, this meant that no matter how impressive her work was, no matter how clearly her colleagues could see its quality, the standard pathways of academic advancement simply did not open for her in the way they would have for a male scientist with an equivalent record. She was praised and respected and then passed over. Again and again. This is the context in which her persistence has to be understood. She was not just sustaining herself against scientific skepticism. She was sustaining herself against a system that was not designed to accommodate someone like her at all. In the American academic world of the first half of the 20th century, women scientists faced systematic exclusion from permanent faculty positions at major research institutions. The science departments of leading universities were overwhelmingly and deliberately staffed by men. Even women who produced work that was recognized as exceptional by their male peers found themselves unable to convert that recognition into the institutional security of a permanent appointment. Barbara spent several years at Cornell in the precarious status of a research fellow. She was doing work that genuinely impressed her colleagues. But a professorship never materialized. She applied for openings at several universities. The responses either never arrived, or came as polite notifications that the position was not considered appropriate for a woman. She held a temporary assistant professor position at the University of Missouri for a period. She continued her corn research there and produced valuable work. But the political dynamics of an academic department made her deeply uncomfortable. She was not constitutionally suited to institutional maneuvering. She wanted to do research and nothing else. In 1941, she joined Cold Spring Harbor Laboratory on Long Island in New York. This research institution gave her what she had been searching for, a permanent place to work, with no heavy teaching obligations, no departmental politics, and genuine institutional support for long-term independent research. Just her laboratory tools, and her cornfield. Cold Spring Harbor became her home for the rest of her professional life. There, she continued studying corn. She planted row after row, observed season after season, year after year. In that cornfield, over those many accumulated years, she developed something that can only be described as an intimate relationship with her research subject. She once said that each corn plant has its own individual character. She could feel them. That was not mysticism or poetic exaggeration. It was the deep practical intuition that comes from genuine sustained attentiveness over many thousands of hours. When you have spent enough time truly looking at something, your brain develops the capacity to make rapid and accurate judgments that appear to arrive from somewhere below conscious reasoning. Some people call this intuition. What it actually represents is a highly refined form of pattern recognition, built up over years of careful and engaged observation. It was in this state of deep attentiveness that she eventually saw what no one else in the world had managed to see. Between 1944 and 1950, Barbara observed a completely unexpected and puzzling phenomenon in the chromosomes of her corn plants. Corn kernels sometimes displayed strange and irregular patterns of color. A dark purple background flecked with patches of white or orange or yellow. The patterns varied from plant to plant, from kernel to kernel, in ways that followed none of the statistical rules of Mendelian genetics. According to Mendel, the expression of dominant and recessive genetic traits should follow predictable and reproducible ratios across successive generations. What Barbara was observing instead looked almost random. It was as if something unpredictable was occurring inside the genome, making modifications to genetic instructions in ways that no existing framework could accommodate. A typical scientist encountering this anomaly might reasonably have assumed it was experimental error or contamination and moved on. Barbara did not see it that way. She carefully tracked the complete parental lineage of every single kernel. She recorded the genetic background of every plant in her growing system. She cross-referenced every observed outcome with what she could see directly on specific chromosomes under the microscope, matching results to precise physical locations on identifiable chromosomes. She conducted crossing experiments across dozens of generations, building up a massive and internally consistent data set. This entire process took her 6 full and productive years of concentrated work. At the end of it, she arrived at a conclusion that she herself initially found almost too strange to accept. Genes move. Genes can physically jump from one position on a chromosome to another. This movement is not random or meaningless. When a genetic element moves to a new location, it changes the expression of nearby genes. This was not a speculative hypothesis. Barbara had built up 6 years of meticulous chromosome tracking records. Every claim she was prepared to make was supported by direct microscopic observation and consistent genetic data. She called these movable genetic elements transposable elements. She identified 2 distinct types of interacting elements with different roles in the system. One type functioned as a controller, determining when and where movement could occur. The other type was the element that actually relocated itself. She named these elements activator and dissociation. The deeper implication was genuinely radical. If genes are not fixed on chromosomes in stable positions, then the foundational assumptions about genetic stability that underpinned the entire field needed to be fundamentally reconsidered. Gene expression might be governed by mechanisms far more dynamic, responsive, and complex than anyone in the field had imagined. Today, with the benefit of decades of subsequent evidence, this seems like a natural and even obvious insight. But in 1950, proposing that genes could jump around on their chromosomes was about as radical as standing up and announcing that the Earth was not the center of the solar system. The prevailing view held that genes were stable and fixed entities. Each gene occupied a specific, permanent position on a specific chromosome. They did not wander. They did not relocate. They sat in their designated places. Barbara was saying the books move by themselves. In 1951, Barbara presented her findings to her colleagues at the annual symposium at Cold Spring Harbor. She came thoroughly and meticulously prepared. She brought detailed data sets, complete chromosome maps, and 6 years of systematic observation records. The response was very nearly total silence. A small number of people raised questions, most of them dismissive. The majority of the audience departed looking confused. Some colleagues concluded that her data must be flawed. Others argued that whatever was true of corn could not transfer to other organisms. And some simply refused to entertain the possibility that genes could move. It contradicted too much of what they already believed. Throughout the entire decade of the 1950s, Barbara's theoretical framework found essentially no acceptance. She attempted to write up the research and submit it to major journals. The responses she received were either confused or simply rejections. There was no sustained engagement, no substantive debate. Her claims were noted and then set aside. The years of the 1950s must be understood not just as professional frustration but as a kind of sustained existential test. Barbara was not facing opposition from a small group of difficult colleagues. She was facing the organized indifference of an entire scientific generation. The people who attended her talks and then returned to their own work as if she had not spoken were not acting maliciously. They were simply unable, with the conceptual tools available to them, to integrate what she was proposing into their existing frameworks. This is one of the hardest things to convey about her situation. It was not that people thought she was wrong in a specific correctable way. It was that they could not find anywhere to put what she was saying. Her discovery did not fit the structure of the conversation that genetics was having at that time. And so it was simply set aside, not refuted, not debated, just set aside. She eventually stopped submitting papers on this subject to major journals. Every submission disappeared without any meaningful response. The isolation of those years deserves to be described with some precision, because it was not simply the experience of being disliked or criticized. Criticism would have been something to engage with. What Barbara faced was more deflating than criticism. It was absence of engagement. When you propose something genuinely new and it is met with sustained confusion and dismissal, you face a particular kind of psychological challenge. The evidence you have gathered does not change. Your understanding of what you have found does not change. But the gap between what you know and what the world is willing to acknowledge can become a kind of daily presence. You wake up every morning knowing something that your entire professional community is not yet ready to see. Some scientists in Barbara's position might have been driven toward bitterness or toward a desperate search for allies and validation. Barbara took a different route. She seems to have genuinely internalized the idea that the timeline of recognition and the timeline of discovery are simply different things. A discovery is real when it is real, not when it is recognized. Recognition follows its own logic, shaped by the readiness of an era, by the availability of tools to confirm new findings, by the accumulated conceptual preparation of a field. None of that readiness could be hurried. All she could do was keep the record clean and complete, keep the work alive, and wait for the world to arrive at the place where her work was already standing. She waited. She kept working. She kept planting corn. Imagine the position she was in. Imagine sharing the most important discovery of your life with your entire professional community. Then watching the scientific world simply ignore it. Imagine doing that, and then returning to your cornfield the next day, and continuing the work. Barbara did not collapse. She went back to her corn. She continued her research. She kept recording her observations, building the record year by year. She later said that if you genuinely know your research is valid, the noise from outside does not actually reach you. You simply continue doing what you need to do. The research itself is what matters, not what others happen to think of it right now. If the research is real, the world will eventually catch up. Those words are easy enough to say in retrospect. To actually live by them, through year after year of being systematically ignored by your entire professional community, requires a quality of inner clarity and resilience that is genuinely rare. Barbara had it. In her small house at Cold Spring Harbor, she continued her life on her own terms. Up early to tend the corn. Afternoons at the microscope. Evenings reading and thinking. Year after year, just like that. Her life was extremely simple. Little socializing beyond the people she genuinely valued. A cornfield and a microscope were sufficient. From the late 1950s onward, the age of molecular biology arrived with transformative force. Scientists began studying genetic mechanisms in bacteria and viruses using powerful new biochemical tools. The French scientists Jacques Monod and François Jacob discovered an elegant mechanism for gene regulation in E. coli that became famous as the Operon model. They demonstrated that gene expression could be systematically switched on and off by specific regulatory molecules. Interestingly, this discovery bore a striking conceptual resemblance to what Barbara had described in her controlling elements framework more than a decade earlier. Monod and Jacob received the Nobel Prize in Physiology or Medicine for this work in 1965. But the molecular biologists of that era still did not draw the connection between their findings and Barbara's earlier work. She had been studying a plant. They were working on bacteria. The disciplinary divisions kept two parallel sets of insights isolated from each other. By the late 1960s, things began slowly but unmistakably to change. Scientists began finding movable genetic elements in an expanding range of organisms. Fruit flies, yeast, bacteria, and eventually in mammals. The evidence was accumulating from too many independent directions to be dismissed any longer. People began going back to read Barbara's papers from the early 1950s. And they began to understand, with increasing clarity, that she had been essentially right about all of it. These movable genetic elements received the universal name of transposable elements, or more informally, jumping genes. Through the 1970s, molecular biology tools advanced to the point where scientists could observe and track transposable elements directly at the level of DNA sequences. The phenomena that Barbara had painstakingly reconstructed through years of indirect observation could now be directly verified. The scientific community began formally acknowledging that what Barbara had described in the 1950s was not only correct but fundamental to understanding how genomes actually work. Today we know that jumping genes exist in every known living organism without exception, including human beings. Roughly 45% of the entire human genome consists of sequences derived from various transposable elements. These elements have played a profound role in biological evolution, influencing gene expression, reshaping chromosomal architecture over millions of years, contributing to the diversification of species, and even participating in generating the extraordinary diversity of the immune system. Scientists have also identified roles for transposable elements in the development of certain cancers and in certain inherited genetic disorders, making them an area of active medical research. What Barbara said in 1951 needed 30 years before the scientific world was ready to fully understand and accept it. In 1970, Barbara received the National Medal of Science, one of the most prestigious honors the United States government awards to scientists. In 1971, she was elected to the National Academy of Sciences. In 1981, she received a MacArthur Fellowship, widely known as the Genius Grant. This prize is awarded each year to a select group of individuals who have demonstrated exceptional creativity and achievement. It comes with a substantial unrestricted financial award, designed to give recipients the freedom to pursue their work wherever it leads. Barbara's name at last began to be widely recognized. Researchers began systematically revisiting the body of work she had accumulated over decades. More and more people came to understand just how far-reaching and foundational her contributions had actually been. In 1983, the Nobel Prize in Physiology or Medicine was awarded to Barbara McClintock. The citation honored her discovery of mobile genetic elements. She was the sole recipient that year. She was also the first woman to receive that particular Nobel Prize as an individual winner, without dividing the honor with any co-recipients. That year, she was 81 years old. 32 years had passed since she had stood at Cold Spring Harbor in 1951 and delivered that presentation that her colleagues had largely been unable to understand. When the Nobel Committee called to notify her, she was out in the yard at Cold Spring Harbor, picking up walnuts. She answered the phone, confirmed the news, said thank you, and went back to picking up her walnuts. That is Barbara McClintock. After receiving the prize, she gave numerous interviews. Everyone wanted to understand what those 30 years of being ignored had actually felt like from the inside. Her answers consistently surprised people expecting a narrative of suffering and vindication. She said she had never felt abandoned. She was simply doing what she felt she needed to do. Whether others recognized it or not did not alter what the research actually meant. When asked directly whether she felt resentment toward the scientific community that had dismissed her work for so long, she said there was nothing to resent. They were simply not yet ready to understand what she had found. Every era has its own limits. That is not anyone's personal fault. She said that if you love your work, you are already a fortunate person. Because you have already received your most important reward. Whatever recognition might come afterward is something extra, something added to something that was already complete in itself. That quality of equanimity is not the same thing as indifference. It is the specific freedom that grows from genuinely and deeply loving what you do. When the meaning of your work does not depend on whether other people affirm it, it becomes very difficult for the world to defeat you. After receiving the Nobel Prize, Barbara's daily life barely changed. She still lived in the same modest house at Cold Spring Harbor. She still went to her cornfield. She still worked at her microscope. She still kept her own particular hours and rhythms. When someone asked her whether winning the most prestigious prize in science meant she had some dream she finally wanted to fulfill, she said she only hoped she could continue doing research. And she did. All the way through her later years, she continued working. In September 1992, Barbara McClintock passed away in New York at the age of 90. She never married. She had no children. Her life, in the simplest account, was her research. But to say only that would be a meaningful misrepresentation. Barbara had deep and lasting friendships with people she genuinely valued, spanning her entire professional life. She loved music. She loved spending time outdoors. She loved walking along the harbor at Cold Spring Harbor. She had a philosophical orientation toward life that was genuinely rich and considered. She once said that the most important thing in life is to find what you truly care about, and then remain loyal to it, regardless of what happens outside. What she left behind is a theoretical and empirical foundation that continues to generate new scientific understanding. The scope of that legacy is worth dwelling on for a moment, because it extends in directions that Barbara herself could not have fully anticipated. In the field of evolution, jumping genes have completely reshaped how scientists think about the mechanisms by which genomes change over time. For much of the 20th century, the dominant model of evolutionary change was gradual, small mutations accumulating slowly over vast periods. Transposable elements introduced a different possibility. They can cause large-scale genomic reorganization relatively quickly. They can move entire functional sequences from one genomic context to another. They can bring new regulatory elements into contact with existing genes. In the language of biology, they are agents of genomic creativity. In the field of medicine, the story of transposable elements is still being written. Researchers have found that certain transposable elements, particularly a class called line 1 elements, can reactivate in cancer cells and contribute to tumor progression. Other elements play roles in the regulation of genes that are critical during embryonic development. Understanding the behavior of these elements under normal and diseased conditions has become an important frontier in medical research. In the field of biotechnology, the conceptual framework that Barbara established has contributed to the development of tools for genetic manipulation. The principle that genetic elements can be engineered to move and insert themselves at targeted locations in a genome underlies some of the approaches being explored in modern gene therapy. And perhaps most remarkably, we now know that a substantial fraction of what was once dismissively called junk DNA, the large expanses of the human genome that do not code for proteins, consists of the remnants and active descendants of transposable elements. These sequences, far from being meaningless filler, are deeply involved in regulating how and when other genes are expressed. Barbara looked at a handful of miscolored corn kernels and saw the outline of all of this. She was just a few decades ahead of the tools and the readiness needed to see it. Research on transposable elements remains a central and active topic in genetics, evolutionary biology, and medical research. Her work fundamentally shaped how we understand genome stability and genome dynamics, how we think about the mechanisms of evolution, how we approach cancer biology and genetic disease, and the theoretical foundations upon which modern gene editing technologies were built. It is no exaggeration to say that every scientist working on genomes today is building on a foundation that Barbara helped to lay. Her story leaves us with a few things worth genuinely sitting with. The first is about the relationship between truth and the timing of when an era is ready to receive it. Barbara's discovery did not become true 30 years after she made it. It was true from the moment she first understood it. The era simply was not ready. Every period in history has its own cognitive horizon, its own assumptions about what is possible and what is not. Some discoveries arrive before the conceptual tools and the cultural readiness exists to receive them. This is a pattern that repeats throughout the history of science. Many times, people who are far ahead of their moment look completely indistinguishable from people who are simply lost. But those two situations are not the same thing. The second is about where we locate the validation of our own worth. Our culture trains us to measure our value primarily through the evaluations of others. Grades, recognition, approval. We are trained to seek these things and to feel their absence as a deficit. Barbara spent her entire life demonstrating that another relationship to this question is possible. She placed the meaning of the work itself above external recognition. Not as a performance of indifference, but as a genuine and considered orientation. This does not mean that recognition is unimportant. It means that if whether you receive recognition is what determines whether you continue, you will inevitably stop at some critical moment. Because recognition is unreliable. It comes and goes depending on timing, on fashion, on who happens to be paying attention. Barbara's confidence was built on something more durable. It was built on her direct and personal understanding of what she had actually found. That was something no one could remove from her. Today we have shared the story of Barbara McClintock. A scientist who persisted in silence for 30 years. A person who forced an entire field to completely rethink what genes actually are. A woman who, at the age of 81, upon learning she had received the Nobel Prize, went back to picking up walnuts. Her story tells us that truly important work does not need an audience to be real. It only needs you. And that cornfield. If today you are doing something that no one around you understands. If your ideas are ahead of where your time has arrived. If you are quietly doing the work in a place with no applause. Think of Barbara. Think of that cornfield. Think of the phone call that came 32 years later. Keep planting your corn. Thank you for being here with me today. See you next time.